Students, today we'll study chemiosmotic hypothesis. So chemiosmotic hypothesis, it explains the process of ATP synthesis. ATP कैसे synthesis होता है ये process explain करता है in chloroplast. So we can write chemiosmotic hypothesis it explains the process of ATP synthesis where in chloroplast. So movement of electron generates a proton. So when electron move, what happen is generate a gradient, proton gradient, and this gradient is used to synthesize ATP. Okay, जब electron move करता है तो it releases energy. When electron release move from high energy से lower energy, it release energy. So that energy is used to synthesize ATP. And this gradient is used to synthesize ATP. Okay, now let's consider a diagram of thylakoid. So this is outer thylakoid. Inner thylakoid. And in outer, what is there? NADP reductase. This complex NADP reductase, and inner thylakoid there is a complex for splitting of H2O. Complex for splitting of H2O, and this is lumen. Okay, thylakoid ka lumen, and here it is PS second, and here it is PS1, and electron acceptor cytochrome. This is, here it is cytochrome. And just clear. It is cytochrome, okay? And what else is here? It is F0 particle. We'll explain in detail. And F1 particle. So this is F0 and this is F1 particle. So we have done all the labeling. Now what happened in this? The sunlight comes, here, ps second. Okay, now electron is released and it will be accept. यहाँ पे electron release हुआ, a pair of electron is released. अंदर ही हुआ है. A pair of electron is released. Okay, and this electron is taken by cytochrome. So during this, what happened when electron is moving from higher to lower energy, तो energy is released. So ये जो energy है वो cytochrome use करेगा 2H plus को, it will absorb 2H H plus from stroma to lumen. One more, this is stroma, this is stroma. Okay, and let me write here 2H plus. So electron is released, sunlight electron is released, to be taken by cytochrome, and when electron move from high energy to low energy, energy is released, and this energy is used by cytochrome to take 2H plus ion from stroma, ये stroma है, to lumen. Okay, so cytochrome here we can write down energy release when electron move from high to lower. This energy is used by cytochrome to take 2H plus from proton from stroma to lumen. Students, this is, I am writing my own words. Okay, just so we can say cytochrome here act as electron acceptor and electron transporter. Now next is, तो cytochrome ने use की, this energy by cytochrome. Now what happened? We can see here photolysis of water takes place. तो जब photolysis होता है, let me write down heading, photolysis of H2O. कहाँ हो रहा है? In lumen. Okay, here it is the complex for splitting of H2O, but it takes place in lumen. So what happens? Three things are released. जब H2O का photolysis हुआ, what product we will get? A pair of electron. और क्या रिलीज होगा photolysis of H2O में? H plus ions. Proton is released and pair of half O2. And what will happen to this a pair of electron? It will be given to, where it will go? Photosystem 2. See, electron release हुआ ना यहाँ पे. And what about H plus ions? It will go in lumen. Go in lumen, or you can say transport in lumen. and make the concentration of H plus high. H plus प्रोटॉन की concentration ह्यूमन में हाई हो जाएगी because of the photolysis of H2, and oxygen diffuse out. It will be diffuse out as byproduct. Diffuse out as byproduct. Okay. So we can conclude here कि ल्यूमेन में H plus concentration ज़्यादा है. How? It is I. In lumen, concentration of proton is high. Why? Because photolysis of water we have seen go in lumen. Second, यहाँ से यहाँ आया ये with the help of cytochrome. क्या लिख सकते हैं हम लोग? मूवमेंट ऑफ इलेक्ट्रॉन, यू कैन से फ्रॉम स्ट्रोमा. Due to movement of electron. Okay. Now what will happen? Here we can say it's F0 particle, F1 particle. So they will help in diffusing H plus from lumen to stroma. यहाँ H plus की concentration बहुत हाई हो गई. F0 F1 particle क्या करेंगे? It will help in diffusing H plus ions to stroma. So we can write F0 F1 particle help in diffusing proton from lumen to stroma. And what is F0? It is an integral embedded protein. Write down integral embedded protein. And F1 is it helps in facilitating diffusion of H plus, or helps in facilitating diffusion of H plus ions. And what is F1? It is intrinsic extrinsic protein. Now what happened? This we have done. Now F1 जो है, when F1 क्या करता है? जब H plus यहाँ से यहाँ आ रहा है, when H plus ion diffuses out stroma, तो energy is released. So F1 use that energy in adding phosphate ions with ATP to form ATP. So we can say F1 use energy of H plus ion when diffuses out stroma. ठीक है. जब H plus ions यहाँ से यहाँ आता है, तो this F1 वो एनर्जी यूज करके ATP को ATP में कन्वर्ट कर देते हैं. तो वी कैन राइट, जब ये डिफ्यूज होगा, इट मीन्स एनर्जी इज रिलीज. 2H प्लस यहाँ आया तो एनर्जी रिलीज हुई. F1 ने क्या किया? फॉस्फेट आयन को ADP के साथ कंबाइन किया एंड इट विल गिव्स राइज टू ATP. And here enzymes ATPase is found. Okay, in short, we can say this is outer thylakoid, inner thylakoid. Electron, when photon is absorbed, electron is given out. It is accepted by cytochrome, and during this, energy is released due to gradient, and this energy is used by cytochrome. वो साइटोक्रोम ने वो एनर्जी यूज़ करके स्ट्रोमा में जो पेयर ऑफ प्रोटॉन थे वो एब्जॉर्ब करके इसको वो ल्यूमेन में ले आए. दूसरा, वह तो साइटोक्रोम एक्ट एज इलेक्ट्रॉन एक्सेप्टर, इलेक्ट्रॉन ट्रांसपोर्टर. Then photolysis of water takes place. What happen? Electron is released. It will finally go to PS second. 2 इलेक्ट्रॉन फिर यहाँ आ जाएगा. एंड H प्लस आइंस कहाँ जाएगा? ल्यूमेन में. एंड ऑक्सीजन विल बी गिवन आउट. तो यहाँ पे ल्यूमेन में H प्लस की कंसनट्रेशन बहुत हाई हो गई. नाउ व्हाट विल हैपन? The F0 F1 particle help in diffusing out H plus ion from lumen to stroma. ठीक है, help in diffusing H plus from lumen to stroma. F0 क्या होता है? इंटीग्रल में हेल्प इन फैसिलिटिंग डिफ्यूजन. और जब H प्लस यहाँ से यहाँ आते हैं तो F1 वो एनर्जी यूज़ करके PI को ATP कन्वर्ट करके ATP सिंथेसाइज कर लेते हैं. ओके, एंड व्हाट विल हैपन टू इलेक्ट्रॉन एंड H प्लस आयंस? इट विल कंबाइन टू NADP and it will gives rise to NADPH2, which is a reducing power. Let me write. Electron and it will be taken by NADPP, and NADP will give NADPH2, that is reducing power. So students, this is all about chemiosmotic hypothesis.